Good evening, everyone. Welcome to another free session on Botany Insider's live stream. Let's wait for the students to join in. Then we'll start off with the class. We'll be discussing about further topics. Yesterday also we revised few topics from unit number 1 because as we know, our December examination is round the corner. So revising the things in a very structured and in a very proper detailed manner is of utmost importance. So I'm trying my level best from my end to provide you a very good helping hand, right? So just let me know whether the audio and the video is clear to you or not in the chat section. Everyone, just confirm whether the audio and the video is clear or not. Then we'll start off. It's a free revision course which will proceed for complete 1 month till your main examination. Examination dates are 26 to 28. At which date do we have life sciences? We'll get the notification soon for the same. Right? I hope everything is working fine. Perfect. Thank you, Harshit. Let's begin with the class now. So, before starting the video, if you're targeting or if any one of you is targeting June 2024 examination, then we have a full course that started this particular week itself for the full June 2024, where live and interactive classes are there. You have hard copy notes, all recordings available, daily practice questions, PYQ 10 years will be solved, regular doubt session, weekly test, etc. All these things in a single course at very affordable prices. So those of you who wish to enroll in this particular course, or those of you who are targeting out the June 2024 examination, you can get yourself enrolled in this structured all-in-one course, right? In addition, for the December 2023, we have a mock test series going on. We have a PYQ session going on for you to practice out things properly, for you to learn out things properly, and for you to get through with the concepts in the best manner, right? So, all the information, all other detailed information you can check on our application. You can download the application from the App Store. You can download the application from the Play Store, both of them iOS and Android available. So, whichever you wish to, you can download the application, right? Let's begin. So first, let's revise about the cleaving agents and their site of cleavage in the protein digestion, because in the last 3 years we are getting questions from this particular area. So it's a very important topic to remember. It's a very important topic to keep a note of, right? Cleaving agents and the site of the cleavage. First one is your trypsin. So we have a number of enzymes responsible, and all these enzymes work in a very specific area for the same. For example, trypsin works on the, or it cleaves the bond between, or when we have arginine and lysine on the side, on the carboxyl side, lysine and arginine, but the adjacent one should not be proline, right? So for trypsin, the workforce area, or the area where it will be working is, let's say this is your N terminal, this is your C terminal, let's say this is lysine. So your trypsin will be working on the C terminal, that is, my trypsin enzyme will be cutting the bond over here, knowing the fact or remembering the fact that adjacent one, the enzyme, the amino acid adjacent to lysine should not be proline. Okay, they should not be proline. If the condition is satisfied, we'll be having the trypsin cutting the bond over here. First thing, in addition, for the same, arginine also works in the similar manner. So remember about trypsin. Then for pepsin as well. Now, for pepsin, the amino acid required, or the area where the amino acid will be cleaving, is your aromatic amino acid, which includes tryptophan, phenylalanine, and tyrosine. These three amino acids are there. Now, adjacent one should not be proline over here. Again, a condition which need to be fulfilled. Then you have the cyanogen bromide. Cyanogen bromide cuts the bond on the carboxyl side of the methionine amino acid. So when methionine amino acid is there on the carboxyl side, it will be cutting the bond. It will be cleaving the bond over there. Then you have thrombin. Thrombin cuts the bond on the carboxyl side of the arginine. Again, important. Then you have elastase enzyme. So elastase enzyme cuts on the carboxyl side of alanine, glycine and serine. Again, condition adjacent one should not be proline. Last is chymotrypsin. Chymotrypsin cuts on the carboxyl ends of your aromatic amino acid residues, knowing the fact or remembering the condition that the adjacent one should not be proline, right? It can cleave ester and the amide bonds both. So remember trypsin, pepsin, thrombin, cyanogen bromide, elastase, and the chymotrypsin. Very, very important. In addition to that, these all are endonucleases. Okay, we have two of the classes for the same. One is an endonuclease and one is an, okay, let me write it here itself. So when we look into the enzymes, so in terms of your peptide digestion, in the peptide digestion, the enzymes could be either endopeptidase or they can be exopeptidase. Endopeptidase and exopeptidase. Endopeptidase are what? The one that cleaves the bond in the middle of your peptide chain. Exopeptidases are the ones which cleaves the bond on the ends or on the final terminal end. So these are what exopeptidases. In exopeptidases, one of the example is carboxypeptidase. What was the example? You have carboxypeptidase as one of the example. These are all exopeptidases which cleaves where? Which cleaves at the C terminal. Where does it cleave? It cleaves on the C terminal of the amino acid. So remember, you have carboxypeptidases working on the C terminal. In the carboxypeptidase, you have carboxypeptidase A, B, and C. Three classes of carboxypeptidase are there, which will be cleaving the bonds on the C terminal. In June 2023, there was a question asked in the CSIR related to the same. Hello, hello, hello. Am I audible? Am I audible? Just check, please. I think there was some issue with the there's some connectivity issue. Just check. Okay. Just wait. I guess there is some issue. Not able to change the slides. Did- Yeah, it's working now. Perfect. Okay, let's continue. Really sorry for the disturbance in the middle. There was some technical issue. Okay, let's continue. So we were discussing about the cleaving agents and the site of the cleavage, right? First one was trypsin, then you have pepsin, then we had thrombin, cyanogen bromide, elastase, and then chymotrypsin. In addition to that, we were discussing about when we learn about the peptidases. So there are two types of it. One is exopeptidase, the other one is endopeptidase. Exopeptidases are the ones which cleaves on the ends. One of the common example is carboxypeptidase. In carboxypeptidase, we have class A, class B, and class C genes. Carboxypeptidase in CSIR in June 2023, there was a question asked related to the same. So you have to keep a note. Each of these requires, like some of them requires cofactors for their functionality. Some of them do not require. For example, your carboxypeptidase C requires zinc. Carboxypeptidase A requires zinc as a cofactor, but carboxypeptidase B does not require zinc. Similar thing was asked in the examination. Okay. So remember about this. This concept is very, very important. And if I show you a question related to the same, okay, now let's try solving out or let's revise about this particular question. If you can recall, yesterday I made you explain a bit about the same. So how many hydrogen bonds involving the backbone of CO and NH can be observed in an alpha helix consisting of 15 amino acid residues? Question is, if you have a 15 amino acid residues, complete polypeptide chain, how many hydrogen bonds involving a backbone of CO and NH can be observed in the alpha helix? Try out the question, everyone. Simple question, important question, and this got repeated in the examination, I guess, the third time. So, try out the question, everyone. There's a very simple basic formula involved in this. Try it out and then I'll give you the formula and we'll be discussing about this particular entire thing. Right? Try the question, everyone. It's a very simple question. You just have to put in one simple formula and you'll be easily able to. Write out, everyone. Question is, how many hydrogen bonds are required involving a backbone of CO and NH can be observed in the alpha helix? consisting of your 15 amino acid residues. So in this particular question, there's a formula which is required, which is for finding out the number of hydrogen bonds in an alpha helix. The formula is n minus 4, where n is equal to the number of amino acids. Okay, this particular question is from June 2022. Yes, Harshit, answer is option number B. So amino acids n minus 4. So 15 minus 4, you get the answer as 11. Very simple question. You should remember that for finding out the number of hydrogen bonds in alpha helix, formula is n minus 4, where n is equal to the number of amino acids. Over here, the number of amino acids given to you are 15. So 15 minus 4, which is equal to 11. So B is the correct answer for this particular question. Easy, important, and scoring question. It was a part B, 2 marker question, right? Remember about this. I hope it's clear. Now, let's revise, or let's try to solve out this question. This was again a repeated question in 2023 paper itself. Question is, in the crystalline NaCl, how many chloride ions surround each of the sodium ion? Question is, in crystalline NaCl, how many chloride ion surround each of the sodium ions? First option is 4, second option is 6, third option is 8, fourth option is 10. So, 4 options are there: 4, 6, 8, and 10. Very, very simple question, easy question, interesting question. Try out, everyone. If you can solve out, please mark the answer. Otherwise, I'll make you explain the same. Yes. How many, in the crystalline nature, how many sodium chlorides surround each of the sodium ions? So, for this particular question, you should be able, or you should remember about the crystalline characteristic property of NaCl, that in the crystalline NaCl, the coordination number is 6 for both sodium and NaCl. The coordination number is 6. So NaCl also and Cl, or Na also and Cl also, both of them has coordination number 6. That means when we look into their crystalline structure, what do we have? Let's say this is the crystalline 3D structure, and if you make up a 3D structure for the same, it's very, very structured, it's very, very organized. Every sodium will be bounding to every sodium will be surrounded by 6 of the calcium, and 6 of the chloride, and every chloride is surrounded by 6 of the sodiums. It's a very, very important question. It's a very easy question also, and very scoring. It's a part B, 2 marker question, right? So remember about this particular thing as well. Answer is option number B. One more question, let's solve out this one, related to the half life. This is again part B question of the recent year. Question is, the radioactive isotope of an element has a half-life of 100 hours. How many hours will it take for 15 upon 16 of the source amount to decay? It's a logical, simple, easy question. Question is, the radioactive isotope of an element is having a half-life of 100 hours. You need to find out how many hours will this particular element take up in order to break down, or in order to decay 15 upon 16 of the entire amount. Consider the unit amount or the initial amount as one basic thing, and then try to solve it out. 1 minute, try it out. Yes, put in your basic maths over here. It was a 2 marker question that was asked. Radioactive isotope of an element has a half life of 100 hours. So, if initially there are two ways of solving it out, either you put in your uh, formula and everything and then try to solve it out. Other way around is try doing it with the help of unitary method. It will be easier for you to solve out. Very good, very good. Others also try it out. Everyone try the question, then we'll solve it out together. It's not a difficult question. It's a logical question. You need not to apply any formula or anything. Very good. So initially I have, let's say, the unitary with the unitary method, 1 unit, right? For half of it to decay, 100 hours will be used. For other 1 by 2 of the content getting degraded, that means 1/4 will be remaining. Other 100 will be utilized. Next, 1 upon 8, again 100. Then 1 upon 16, again 100. Question is how much time or how many hours will it take for 15 upon 16 of the amount to decay? 15 upon 16 of the amount to decay, or we can say 1 upon 16 amount remaining, right? You can solve it with back induction method also. Either 15 upon 16 source decayed, or 15 upon 16 of the content decayed, or 1 upon 16 content remaining. The second method is much easier for us to solve. So you have 100 plus 100 plus 100 plus 100, 400 hours will be required, or at 400 hours you have 1 upon 16 fraction of the content remaining. So B is the correct answer for this particular question. Very, very easy, very, very important, very logical. You don't even require a pen and a paper for solving out such a question. If you have a very, very simple grip over mathematics, then also you can easily solve it out. Is it clear to all? Yes. 8 of you are there in the class. Is it clear to each one of you? Just check whether it's clear or not. If yes, then we'll solve out few more questions, because during the end time when the examination is around the corner, revising or practicing out the questions are very, very important. Perfect. Let's move forward. Let's solve one more question. Right, we have discussed about this. This is one of the segment. This is the next segment. Now you have line weaver plots for inhibition in CSIR. This particular segment gets asked a number of times. That is in competitive inhibition, in uncompetitive inhibition, and in the third scenario, the non-competitive inhibition. There are conditions when you have Km and Vmax getting changed or getting either increased or reduced in one way or the other. So you have to remember, in the competitive inhibition, you have Km and Vmax getting, or they remain unaffected. In the uncompetitive inhibition, your Km is reduced and your Vmax is reduced. In case of your non-competitive inhibition, you have Km unaffected and you have Vmax reduced. So this is something that you have to keep in mind very, very nicely, that in case of your competitive inhibition, Km is increased, Vmax is unaffected. In case of uncompetitive, Km is reduced, Vmax is reduced, and in case of non competitive, you have Km unaffected and remarks, reduce, important, easy, and very much repeatedly asked in the main examination, right? Remember about this. Very important and very, very scoring segment, right? Clear everyone till here? Yes. Clear till here? Just check whether it's clear or not. I'm just giving you gist of very small topics. It will help you in understanding the entire thing. And tomorrow there's a video coming up for 32 most important topic list, short topic list. So make sure that you watch it. I'll be uploading it tomorrow. So watch that. It will help you out in keeping your revision on point, right? So these are the things that we have to do today. We'll do the remaining things in the next class tomorrow. Like till your main examination, we'll be having a class every single day. So be there in the class, and these classes are going to help you out in understanding, revising, recalling few small segments which can help you in the main exam also, right? If any of you is targeting the June examination, the June, if you're planning out a proper attempt in the June exam, so we have a full course ongoing for the same. You can download our application. The link is in the description box and get yourself enrolled in the same, right? In addition, just let me know what all other type of content do you want me to take up in these sessions. Whatever is your suggestion, I can take up the same, which can help you out in the maximum or in the best of my capacity. I'll try to help you out till the main examination. Right, so this is about it. All the best, good luck, take care everyone. Let's meet tomorrow now.